Alrighty, hi everybody. This is another Fireside Chat, this time with some of the seniors, including myself, Shauna and Malky, but they'll introduce themselves in a second. So even though I kind of already introduced you, introduce yourselves.
Hi, everyone. My name is Malky Diedman. I'm the president of BAPS and co-president with David. And I'm a senior at Barnard, majoring in neuroscience. And I currently work in a lab in Barnard, and I have been since my sophomore year. So I'm here to answer any questions about that.
Hi, my name is Shauna Gershbaum. I'm the treasurer of BAPS, and I'm a senior at Barnard, also studying neuroscience. And I work at a lab at the Columbia Medical Center. And I've been working at this lab only for this year, but in the past I've worked in a different lab at the Columbia Medical Center for 2 years. So I have different research experience in both like wet lab research and also remote research. So I'm happy to answer any questions about that.
As people watching this may be able to tell, I am currently in a lab at Columbia Medical Center in the Taub Institute for Alzheimer's research. So that's also neuroscience. But why don't we kick it off with the questions that we have here, which is, what kind of research do you do? So I kind of just said mine. I can go into some more detail, but I usually talk a lot every week. So if you guys want to start off, that would be great.
Wait, what was the question? Sorry, you missed it.
It was the first question. It's what kind of research do you do? So I said, since I usually talk quite a bit, if you guys want to start off, you're more than welcome to.
Sure. So I work in a research lab, obviously, in Barnard, and I do clinical research. So I mostly work with mice. I have been since sophomore year, and not clinical research, sorry. And I've been doing I've been testing the same idea since sophomore year, but in different ways. And it's been really helpful starting so early. I don't recommend that for everyone because you kind of get a little burnt out, but it is nice because I do feel really comfortable in the lab, and I got to know everyone, and I got really comfortable with all the instruments and comfortable handling mice. And you probably can if you work there for a short amount of time, but I definitely think there is definitely some advantage to starting early. And also as I do it longer, I definitely start to like it more and definitely learn more. So that's also definitely a pro to it. Yeah.
I started doing research in the middle of my sophomore year, and at that time I worked at the TABOS lab, which is at the Columbia Medical Center, and that was a cardiometabolic lab, and it was super interesting. And also, like Malky works with mice, it was like a very wet lab, and I really enjoyed it a lot, and I learned a lot. And I also think starting early, you could get a little burnt out, but it's good. You have a chance to really develop your skills, and you also have a chance to go from just learning how to do it to also the next year when you're more advanced, you could also think things through, and then you could also start having questions yourself that you could start looking for answers to, which is great. And for this year, I needed to move to a neuroscience lab for my thesis. So I stopped working at the TABOS lab, and now I work at the Calendonc lab, which is a neuroscience lab. And we work specifically with Alzheimer's and Parkinson's disease. We're focusing on those two diseases. And my research has been fully remote this year. So in the past, I'm working off of data that's been collected in the past and using deep learning to analyze that data.
And with my research, like I said before, it's Alzheimer's research, also involving animals. So I've been involved with animal behavioral research since the summer of 2019. And like Malky said, you definitely get pretty comfortable with it, and it gives you a lot of opportunities from what I've found to get relatively good hand skills, and that carries you through your career, obviously. But whether you're doing injections or surgeries of various types, you're getting that exposure that helps you build on things that you're going to need to know later. So it's nice to get kind of do some of the legwork now and get comfortable with things. And another kind of added layer is if you're doing, you know, like an injection, for example, to a human, probably not gonna bite you. So like, you know, you get a little bit of a better handle on things as well, just because of that like added like danger effect. Not that everybody necessarily when they do research does animal research, but it's just like a fun little way of describing it. So looking also at, you know, how we came to get our positions that we got. Um, uh, Maybe you guys also want to start with this one.
Yeah. So I work in Professor Glendenning's lab. It's called his Taste Lab at Barnard College. And I walked in, I was a freshman and I was super eager to start research. So I think that summer of my freshman year going into sophomore year, I emailed like every professor that I like knew of. And he happened like we had a relationship already because Professor Glendenning is my advisor. So then slowly starting sophomore year, I began to get like I took all the wet labs and all that, and then second semester sophomore year I was finally able to like actually work with the mice and start. And also another advantage of starting sophomore year is that I didn't have to like go in like head on and take on everything at once. I was able to like shadow a senior, and like it was really her thesis project or maybe even a bio major, and it was like her project, and I came in on like the days she couldn't, and like she taught me what to do slowly, but like there was no pressure for me. Like I was able to do it kind of like take a backhand seat to it, which was a really great way to learn because I wasn't like thrown in and had to find everything myself. And if I messed up, like I was so upset, like nothing was really my responsibility yet. So it was really nice to start like that. And yeah, that's it. Really, that's how I started in my lab.
I had in my sophomore year, I wasn't as on top of it as Malky, but in my sophomore year, I also was like, okay, I think it'd be a good thing to start getting into research. I wasn't sure exactly like if I was gonna be pre-med or pre-dent, but I knew I wanted something in the health field, and I knew that research could be very helpful. So I was like, okay, time to get started. And I think I emailed one or two people at Barnard, and I think that labs were very, very full and it was tough to get into. And I was on like the Barnard Neuroscience listserv, and someone from that listserv sent out an email that someone at the medical center was looking for a research assistant. I jumped on it immediately. And then it was like a small like application process. I did my resume, but then I had a really great opportunity to meet with this woman who would then become my mentor at the time, meet with her in person. And when I met with her in person for like a small interview, it was my first time going into like a real lab, kind of like what David's sitting in now. And when I met I had a chance to really meet my mentor, and she was excellent. And I think that that's something that's really valuable when looking at a lab, that not only do you want to have, it's very nice being a prestigious lab. And my mentor, she wasn't the PI of the lab. She was a different researcher, and the PI, it was a nice prestigious lab, and it's great to have those connections. But what was really helpful for me was my mentor was fantastic, and she was really great and wanted me to learn. And I think that was really important for me, and that's what I was looking for in a lab. So that was a great opportunity for me, and that's how I got like my first taste of research. I really got to learn a lot, and she was very patient and was willing to talk through things with me and not just tell me what to do, but also really like talk through and see like, do you understand the reasoning behind it? And I think that's very important when looking in a lab, that you want to have someone who's really going to walk you through things, help you through, and want you to understand like the background behind it as well. And then this year when I worked at a lab, I was like in a little bit of a panic zone. And I think I emailed like every single psychology and neuroscience professor in Barnard, Columbia, and at the medical center until someone said yes. And then also same thing again, I just got very lucky that it was a very nice mentor, and same small application process. And then that was it, I was in.
Similarly to what you guys were saying, and this is something that carries through most of my life, is I love a cold email, and I send them to everybody all the time, um, constantly. And so, you know, when it was like halfway through my sophomore year, I was like, well, it's time for me to find research. That's something I need to do. I went on the website for Columbia Medical Center and emailed, I think in 2 days, I probably emailed 80 labs, 80 PIs, and because I just went down the list alphabetically. But the lab I'm sitting in was the one that I was kind of hoping would get back to me and be like, yes. Uh, and so they called me in for an interview, Dr. Arancio, the PI. And he was like, tell me what you know about neuroscience. And I said, I don't know anything about neuroscience. But they took a shot and said, we'll give you a chance. And it worked out really well. I was able to start right after the semester ended in the spring. So beginning of the summer. And I kind of, at first I was doing, like during breaks, so like summer, winter intersession. But this semester I'm here. I structured my schedule around being able to be here at least 5 days a week. Um, and now probably going to be a little more on the weekends as well, because I really, something that, and I'm kind of going on, but like something that I used to say, during the semesters would be like, I love learning and I love classes, but I really wish I were in the lab right now doing lab stuff. So I kind of made it so that my last semester is completely, devoted to lab research, which is really great. And unlike you guys who might have like one mentor like you described, I'm lucky enough to have a whole bunch of people who are kind of mentoring me in different areas of the lab, just because we're rather large. So we have different sections that I'm able to kind of pick up some things in, which is really nice. Um, so yeah, that's kind of how I came to get my lab work, and that's my kind of experience. Um, and then another question that we have that we set up is, what's the point of research and how does it help? Which I think we kind of touched on, but we can go over a little more in detail. So if either of you guys want to jump in.
So there's so many points to research. Well, one point could be like maybe you're actually interested in what you're researching. So I'm researching diabetes and different ways our body releases insulin. So like that definitely is important, and I'm very excited to find the results of my experiment. Another reason is just to like have something to talk about at your interviews and your applications. It's definitely important. You should definitely do things you're passionate about, but it definitely doesn't hurt, and it's good for all that stuff. And specifically, for my research, I had to do surgery on mice. And for dental school, they ask you about like dexterity and different activities you do with your hands. So I found that like right away, like I knew exactly what I was going to talk about. And I also like it makes me realize that I do like doing like little things. Like I like looking through the microscope and all that stuff. So I definitely enjoy that. And I'm just learning new things about science. I'm getting my feet wet in the science world and like hearing the lingo and how people talk about it and how to read papers and how to write papers. So I find all that really helpful.
I think Malky hit like most of the main points, but I think I want to highlight the manual dexterity piece. I think that's a huge thing because we have like a little bit of opportunity in like our labs for classes and things like that to work on it a little bit, but you have a chance to kind of like do it consistently. It's important, and you see that like you improve, like it's a skill that can be improved. So I think that's very cool that to be able to see that and find that when you're doing research. And I think that also a lot of the material that you learn in class, it helps you like understand it more, and like you get to see it come to life. Like I remember learning about optogenetics for the first time, which is like a method that allows you to like turn on and off certain neurons in the brain using light. I understood it very abstractly, but I didn't get it. And then now I'm doing that in my lab, and you just understand it in a whole different way. And it helps you understand, like Malky was saying, like the world of science, like where that's coming from. You get to learn a little bit about like the next steps, how people are thinking, like what research is being done, what research can you do, the literature that's out there. And also if you're lucky, you could also get to be in the process of writing a paper and publishing a paper and get to see how that all comes about. So it really benefits you as a student also to have something to talk about, maybe even have something to publish, which is like a great achievement, and you always have that to your name. And also it just allows you also to make more connections. You can make more connections with different mentors and different things. And there's also a lot of research that overlaps with like dental research. Like the lab I worked at my sophomore junior year dealt a lot with like blood clots and bleeding. And that's a very big area in dental research actually, which I didn't know at the time, with like gums bleeding and things like that. So it's very good also to have those connections and also those things to talk about in the future.
You guys, like Sean just said, hit all the points. One thing I'll say that's kind of maybe a little different is, in in the lab I'm at, we do on Tuesdays in the afternoon, we do a lab meeting where we come together and somebody presents research from another lab that they've found and they've looked at, and they present on it. And that's something that I've been finding pretty valuable because I like to take notes on it and really kind of look back at it and see what's going on. Because even though things are coming to life, in what you're doing every day, getting to look at what other people are doing in their labs and hearing scientists talking about other people's research is really eye-opening and a great way to learn. I think it's a lot more interactive than maybe a big lecture class might be, just because you're with these people, there's not necessarily any pressure to ask questions. You're able to kind of just be completely absorbed with it. So it's a really great that's like a really great perk in my opinion. And looking also at projects. So what kind of projects we're working on? You guys want to jump on that?
Sure. So I kind of spoke about this a little, but basically when you ingest glucose, or mice ingest glucose, their insulin is released before blood sugar rises. So we're trying to see what mechanism in the body causes this early release of insulin. We call it cephalic phase insulin release. So we're testing that by cutting off a way that the glucose goes to the stomach. So we're tying the esophagus shut. And we saw that if we tie the esophagus shut and then pour liquid in the mouse's mouth, the mouse would drown. So now we also do a tracheotomy to prevent the mouse from drowning. And then we fill the mouse's mouth with fluid, either the control, which is like a saliva, or fructose, or glucose, which is the test solution. And I haven't gotten the results yet. But we take blood before we put the solution in the mouth, and then after. And we're going to check insulin levels before and after, and blood glucose levels before and after. And then we'll see if this makes the insulin be released before the blood glucose levels really increase. But yeah, so that's what we're doing. I don't know if that makes sense, but
I'm working on a project where my lab looks at is it neuroscience lab, or working with Alzheimer's and Parkinson's, and we're looking at an area of the brain called the basal ganglia, which is involved in a lot of things, but what we're looking at specifically is voluntary motor movement. And in the past, what they have done in the lab already was they found out that there's these neurons in the brain in the basal ganglia called bridging collaterals. And those neurons help control, they kind of bridge these two areas of the brain. One part of the brain kind of like tells movement to go, and the other part of the brain tells movement not to go. So those neurons connect both of those, and they help regulate voluntary motor movement. And and voluntary motor movement, that's like loss of that voluntary movement is a symptom in Parkinson's disease and also sometimes Alzheimer's. So we're trying to understand the mechanisms behind those neurons and the way that they regulate that movement control. And what we did is we turned on and off those neurons in mice in the brain using optogenetics. And we watched them in an open field, which is just like a very large area, to see the amount of movement when we turned on the neurons and we turned off the neurons. So now what I'm doing is it's super cool. It's like a new up and coming area of neuroscience using deep learning. And we videotaped those experiments, watching the mice run around with certain neurons on and certain neurons off in their brain. And after videotaping them, we're now using this computer program called Deep Lab Cut. And it allows us to train a neural network to analyze all these videos and look on like a really, really fine level, the amounts of movement and how the voluntary motor movement works with these neurons turned on and off.
And I'm kind of working on a few projects. I'm helping some people with certain parts of their projects, running some control testing. And I'm also starting now my own project. So those projects are working with certain new kind of drugs that we're developing that we look a lot at kind of more like genetic kind of part of it. So working with stuff called HAD activators and HDAC inhibitors to see how those kind of interact with neurodegeneration. And so, for example, we're doing a few different things with that, and that's what I'm going into now. And as far as like running behavioral testing, we do a battery of behavioral tests, and we're able to kind of graph out and see a real substantive difference between our our different groups, our different cohorts. So that's been very interesting to see, getting to look at the data analysis or doing the data analysis as well. And then as far as the surgeries that we do, they're neurocannulations, so that we're able to send either amyloid beta or tau proteins, which that's part of what we're doing our research with, using the inhibitors and activators to kind of see how we can play around with those proteins and oligomers. So the surgeries are neurocannulations, so that we're able to send the proteins or whatever it might be straight to the hippocampus. So that's kind of what I'm doing. So doing some other people's stuff and also starting my own thing, trying to get a dose curve is essentially what I'm doing. So yeah. Anyways, so that is the questions that we had written out. Do you guys have anything else that you feel like super excited to say about what you're doing, or anything else that you think is important to know about research, maybe for anybody who's interested in pursuing it?
I just want to say, don't be afraid to switch labs and to try out new things and to see what interests you. Like maybe you don't like working with mice, maybe you do, maybe you want to do more of like a psych research, but definitely try new things and find what you like, because it will definitely be more enjoyable because you end up putting a lot of time into it. So that would be my advice.
And also, I think that there's a big value to being able to do like consistent research. Like I found like during the semester, my hours were so choppy and I was all over the place. And I was able to do it like research consistently one summer. And like I felt that my skills really improved. And like I think that for me, I wasn't always like so natural with my pipetting. It felt a little clumsy at first, and I wasn't like the most precise, but you see yourself improve, and it's a very cool thing. And then like the data that you're doing hopefully like gets published and it's very meaningful and significant, which is a very nice feeling as well.
Yeah, and kind of going off of that, I agree. It's really great to be able to see your skills kind of improve. That's one of the best. It's like a great feeling to be like, I can do X, and that's, so when people ask you, what do you do? You're able to not only explain to them the scientific aspects of it, but also say, I can do, this, and this. I have these skills that I've acquired. And then as far as a piece of advice that I think might be useful is always ask questions, which might seem kind of obvious, but I know that for a little bit at the beginning, I was nervous to ask, like, why do we do this, or what's the meaning of that, just because I felt like it would be, people would think it was silly. But I now ask questions constantly just because, you can't, like, you need to be able to learn things, that's why you're at a lab. So, uh, by asking all the questions you can, it's really an excellent opportunity, taking full advantage of both, the time you have at the lab and also part of the reason why you're at the lab. So.
I want to add one more thing off of that, if that's okay, that you reminded me that asking questions is such a good point. And also, first of all, from a practical perspective, if you're like actually doing something and you kind of don't understand it, but you just go along with it, odds are, one, you'll have to do it again and you still won't get it the next time. And two, like you want it to work out. So that's important to understand. And on another note, it's so cool to see that each area of research, like each area of science has so much material and like so much depth. And like I remember getting to the point where like when you Google the proteins, like the names wouldn't come up, and I was like, oh man, like that's how you know like you're really in it, and like you're really jumping into like a whole area of science that like you don't have so much background in. Like your classes don't prepare you for that because you're not majoring in that one specific protein, but all of a sudden like you could spend years just looking at that protein. So you really should not be afraid to ask questions because no one knows all of that material before going in. You don't have a PhD. How would you be a professional in that area? So definitely ask questions.
All right, well, I think that's about it. It was a quick one tonight, but that's definitely okay. So thank you guys for speaking with me. It's nice to have other people doing some of the talking. uh, and we'll see you guys in the next Fireside Chat on YouTube. See you.